Good morning, everybody. We are Riccardo Smeriglio and
Marco Brate.
we are here to talk about our work on fast and accurate LSTM meta-modeling of TNF-induced tumor resistance in vitro. Multi-level hybrid models are the key to better understand biological systems. In particular, as you can see in the figure at the right, an in vitro model gets modeled as an in silico model in order to allow us to perform operations on this in silico model, in particular performing simulation, to predict the behavior of the in vitro model without wasting resources, time, and money. In particular, this kind of in silico model are very complex, and this is due to the complexity of the in vitro model that they aim to model. And this means that they require a lot of time and a lot of computational power in order to execute a single simulation. In the context of optimization via simulation, that is the process of determining the best configuration of a biological system by optimizing the in silico model instead of the in vitro model, are required several simulations. And this can be a problem in the context of time and computing power, because all of the simulation require a lot of time and a lot of computing power, and to optimize an in silico model using, for example, optimization algorithms are needed a very high number of simulation. In particular, our work aimed to replace the simulator with a surrogate model based on artificial intelligence and neural networks that aim to reduce the time and the computational power needed for executing a single simulation without losing accuracy. In particular, this kind of surrogate model, of course, have the potential to approximate simulations, but also maintaining accuracy and reducing computational costs. In particular, in this context, the neural networks better suited for this purpose are recurrent neural networks and, for example, long short-term memory, because all of these kind of simulation are time-dependent, since they aim to simulate the behavior of the cells or whatever is the in vitro model over the time. So, long and short-term memory networks are well suited for this kind of application, because they allow us to take time series. In particular, we selected a single simulator called Physibos 2.0 that is a multi-level simulator that operates across 3 different levels: the 3D microenvironment that simulates the substances diffusion, the agent-based cellular models that cover cell volume, motility, mechanics, motion, cycling, and death, and allow also to switch from a specific cell type to another cell type, and the intracellular signaling and regulatory network that simulates the cell's internal signaling pathway using Boolean models. This was an add-on to a previous simulator using MA-BOSS. In particular, the selected use case aimed to simulate a tumor necrosis factor administration of a 3T3 mouse fibroblast tumor spheroid. The aim of this administration is to kill the cells, the tumoral cells, and the specific administration of TNF should kill the cells, making them necrotic, while the apoptotic death occur due to randomness or natural death. Of course, a specific treatment can be not useful and not effective, and in this case, the cells remain proliferative in a state of alive cells. So in this context, there are 3 different types of cells that define how the protocol, how the specific stimulation is going, that are alive cells, apoptotic cells, and necrotic cells.
Thank you, Riccardo, for the introduction. I will now go through the dataset that we built to train our surrogate model. And in particular, we selected 3 input parameters that are useful to the Physibos simulator. And these are the pulse period that tells us the time interval between each TNF stimulation, the pulse duration, which tells us the duration of the stimulation, and the TNF concentration, which gives us the intensity of the TNF stimulation to the cells. We then covered, as you can see, a wide range of possibilities for these input parameters while maintaining biological relevance, because these parameters were also used for an OVS by the authors of Physibos. So we executed around 2,000 simulations of 1 day. And of particular importance is that we created separate data sets based on the tumor radius parameter that we used, that goes from 50 to 400 micrometer. And this indicates the initial number of cells used in the simulation. And as Ricardo said, we recorded the number of alive apoptotic and necrotic cells at specific time intervals of 60 minutes throughout the simulation. We then built the architecture of our surrogate model, which is a recurrent neural network using the LSTM module. And this takes three input parameters that we previously defined, which are the pulse period, the pulse duration, and the TNF concentration. These are passed through multiple feed-forward layers and are given as an input to the LSTM through the cell state and the hidden states. To the LSTM as an input are also given the number of alive apoptotic and necrotic cells at each time steps. And the LSTM purpose is to predict what the number of these cells is going to be in the next time step. And this is done through a decoder, which is again multiple feed-forward layers. This is then compared to the ground truth to train our architecture. So a bit more about the preprocessing and training of our model. We split the data into a training set and a validation set. We didn't use a test set because we didn't perform any optimization of the hyperparameter of the model. Also, the number of alive apoptotic and necrotic cells was min-max normalized based on the tumor radius that we used. And finally, we trained our LSTM using backpropagation through time, and we minimized the objective function, which was set to the average mean squared error between the predicted number of cells and the ground truth. And you can see here defined the objective function on the bottom, and on the bottom right, you can see the trends of the training losses and the validation losses for each of the tumor radii that we used. In terms of results, overall, we can see that our model is fast and accurate at reproducing PhysiBOS results. And on the figures on the right, you have one figure for each of the tumor radii that we used, so from 50 to 400. And the trend is the trend of the number of cells, so alive, apoptotic, and necrotic, throughout the simulation. And the dashed line is the predicted number of cells for each of these simulations. And you can see that the LSTM does a very good job at predicting the number of these cells. In particular, the average relative error is around 7.5% across all of the simulations that we tested. And also, the LSTM is 5 orders of magnitude faster than the simulator. A little bit more in detail, in terms of the mean absolute error between the prediction and the ground truth, you can see here on the right a trend of these values, also separated by the tumor radius. And what we can observe is that for apoptotic cells, the error is always very low, except for a little transient in the first half of the simulation for the two smaller tumor radii. On the other hand, for alive and necrotic cells, the mean absolute error increases overall over time. And this is due to the fact that the RNN, being a recurrent model, propagates the error throughout the simulation time. Finally, in terms of relative error, you can see here the relative absolute error between the prediction and the ground truth. And on the right, you have a figure of the error based on the tumor radius. And what you can observe is that for smaller radii, so for example, radius 50, then the relative error is high. And this is due to the fact that even for very small errors in the number of cells, the relative error is very high due to the small number of initial cells in the simulation. But for the simulations with the highest radii, then the error is very low and is below 5% for the two higher radii that we used, for example.
Thank you, Marco. I'm going to talk about the time comparison between the original simulator Physibos 2.0 and our model. Actually, for our model, the training time takes more or less 3 minutes and 30 seconds, while the inference is basically instantaneous. And while in terms of time, for Physibos 2.0 simulator, the time required for a single simulation depends a lot on the number of cells, as we can expect, since all the cells are simulated differently. So with a time radius, with a tumor radius of 50 micrometers, the number of simulation needed to reach the training times needed to train the specific model is more or less 70 simulations, while in the best case scenario with a radius of 400, the time required, the number of simulation required is more or less 13 simulation. This means that our model is able to reproduce the behavior of Physibos 2.0 with 5 order of magnitude faster than the original simulator. And the only phase that is taking a little bit of time is the training time, but is needed in order to have that specific model working. Of course, 70 simulations are not that many in the context of an optimization via simulations, since for optimizing this kind of process, the number of simulation needed is way above 70. In particular, our model was able to successfully and efficiently reproduce the behavior of the simulator in this specific use case. Still, this model lacks of generalization. In particular, the fact that we trained, in particular in the future, we aim to avoid training different models for different radius, but training one single model for all the radius. And moreover, we would like to develop an all-purpose surrogate model capable of handling not only this specific use case, but all the use case in Physibos and in other simulator, independently on the selected simulator and the selected use case. Thank you all for your attention. Here in the figure highlighted, you can see all the author of this paper, but we would like to thank you for your attention, but also to thank all our group, the Smileys group in Politecnico di Torino, for the support and the possibility to work with them. Thank you very much.
Thank you.